So welcome everybody. If you like to have a seat, please take one. So welcome to this year's Senior Thesis Poster Session. This is actually our 10th anniversary, so we have done this 10 times, 10 years in a row. Very proud of ourselves. And it's time to celebrate the accomplishments of our fantastic seniors and celebrate their work, and as the bee does in this little picture, eating the nectar of the flower. My name is Martin Stute, and I'm a professor in the Environmental Science Department, and I'm the sort of coordinator of the senior seminar. But there are 6 other faculty members from the involved departments that help me with this, and I'd like to acknowledge their contributions. There's Professors Bose, Bowman, Palmer, Furman, and Webb. And I also would like to thank all the research mentors that have worked with the students on their research projects. So they have put many, many hours into this, and we really appreciate their support. I also like to thank the Earth Institute that's sponsoring this event and providing the food for us and the drinks for us, and being generally supportive of the students and providing money for summer internships, etc., which is fantastic. So for the next 40 minutes or so, the students that are lined up there are going to give you a 1-minute overview of their project, 1 slide each. They're going to walk up here and then walk down on this side, and you can find a seat or stand there afterwards. And they will present in the order that's given in your program. Hopefully you all have one, so we will just go down that list. The posters afterwards, after these 40-minute presentations, we want to pull the chairs together in the center, so we would appreciate your help to stack some of these chairs. We put them all in the middle, and then we are moving the posters in a little bit because, as you can see, the light in the back of the room is not very good. So let's get started. And one more thing, please. Please hold your applause until the last student has presented, and then we'll have, we'll have a big applause afterwards. All right, let's get started.
DDT was widely used as a pesticide in the latter half of the 20th century. However, it was subsequently discovered that the compound was extremely toxic and could have detrimental effects on the human reproductive and immune systems. Although the production of DDT has been banned or severely restricted in many countries around the world, there still remain measurable levels in the air and soil, largely due to the stability of the molecule. Humans are exposed to DDT through inhalation, ingestion, or dermal absorption, and once inside our body, these compounds associate with our adipose tissue, largely due to the fact that they're extremely lipophilic. Currently, the exact mechanism of mobilization of DDT around our body is not clearly understood. I utilized an animal model to further understand how and where these molecules are mobilized during times of caloric restriction and fat loss. Our results indicate, as you can see, that these compounds target other lipophilic tissues, such as the brain. It's important for us to have a thorough understanding of how DDT molecules are mobilized in order to design an effective treatment. Thank you.
Good afternoon, everyone. My name is Brittany Wilson, and my research examined the effects of obesity on oxalic nitric oxide in mice models. Oxalic nitric oxide, also abbreviated as ENO, is a commonly used biomarker to diagnose asthma because asthmatics tend to have significantly higher concentrations of ENO. My research had 2 goals. The first was to develop a method that would allow us to measure ENO in mice models. And the second goal was to collect and analyze ENO in obese mice and non-obese mice and observe the differences. As you can see on this graph, my research showed that leptin-induced obese mice had significantly lower concentrations of ENO than non-obese mice. And to find out exactly what that means, come talk to me later.
Hello, my name is Eric Nickelman, and my research was looking at the correlation between urinary arsenic and socioeconomic status in Bangladesh communities. Currently, there is about 35 million people who are exposed to arsenic in Bangladesh, and long-term exposure to arsenic has major health consequences, such as heart, lung, kidney, liver cancers, cardiovascular disease, and skin lesions. And in 2000, arsenic exposure was made public knowledge to the community of Arza in Bangladesh. And so we had predicted that in doing this baseline that that the exposure was the same for both the lower and the upper socioeconomic groups. And 2 years later, we found that the reduction in arsenic for those who were exposed to unsafe wells was 109, and for those from drinking from safe wells was 7. So we predicted that those who had the major reduction were from the upper socioeconomic studies. The results showed that there was a significant difference between the exposure between the two groups, and we also found that there was a correlation between high concentration at exposure and the high reduction. Thank you.
Arsenic contamination has been the cause for widespread disease in South and Southeast Asia, diseases such as lung, skin, and kidney cancers. Organic carbon present in the subsurface creates the reducing conditions needed to mobilize arsenic. In this study, intact shallow Holocene and deep Pleistocene sediment columns were used. Artificial groundwater amended with organics was run through 10-centimeter long columns. Results show that organic carbon can be slowed down at the subsurface by as much as 1/16 as slow as water. However, results also show that arsenic in low concentrations can be mobilized by organic carbon. Therefore, albeit slow, organic carbon transport can cause arsenic contamination of deep and shallow wells.
In Bangladesh alone, over 60 million people are exposed to elevated levels of arsenic in their drinking water. This chronic low-dose exposure to arsenic can cause several adverse health consequences, including increased risk of skin and kidney cancer, as well as the impaired motor and intellectual function of children. Previous research has shown that the microbial reduction of iron oxyhydroxides in the subsurface is what leads to arsenic release into groundwater. Our study is looking at whether this carbon that drives the bacteria is from a natural source, such as sediment or peat, or from an anthropogenic source, such as human or agricultural waste. In April 2011, we traveled to Sharasti, Bangladesh, which is the site pictured here, and we filtered groundwater and collected bacteria from 3 different wells, which are denoted by the arrows in the picture. And what we found was that the radiocarbon age of the microbial DNA resembled that of the sediment, which suggests that even at a site that has a lot of anthropogenic inputs because of a high population density, arsenic release can be a naturally occurring process.
Hi, I'm Celia, and for my project, I analyzed seismic wave amplitudes in order to determine properties of the crust and upper mantle in the western United States. While surface wave phase velocities are routinely used in seismic studies to determine structure and composition of the solid Earth, surface wave amplitudes are used much less frequently because of difficulties in making accurate measurements. In this study, I used Rayleigh waves recorded on the US array, a dense network of seismometers placed across the continental United States, to determine local amplification factors at each US array station, which reflect local elastic structure beneath the stations. This map shows local amplification factors for US array stations for Rayleigh waves at a period of 50 seconds. In addition, I compare observed surface wave amplitudes with those predicted by 2 different Earth models in order to assess the ability of current Earth models to accurately predict surface wave amplitudes. Finally, I examined the impact of perturbations in the velocity in the crust on surface wave amplitudes. Results of this study indicate that surface wave amplitudes are indeed a good constraint on elastic structure in the crust and upper mantle. Thank you.
Good afternoon. My name is Andrew Westbecker, and I studied the faulting of the downgoing oceanic plate in the Alaska subduction zone. I wanted to look at the faulting that occurs along the outer rise of the oceanic plate at subduction zones, because it's a primary means for water to get into the plate. It's important to study this hydration because it's integral to the creation of seismic activity, or the creation of magmatic arcs and controls in seismic activity. This last summer, I went with a team of researchers on the research vessel Langseth to the Alaska subduction zone to look at the bathymetry and subsurface structures of the outer rise. We found that oceanic lithosphere with fabric oriented parallel to the trench has its faults with larger offsets. If larger offsets mean more water, then that can be a clue to the seismic and magmatic activity in the area. The fabric orientation of the incoming crust and the orientation of the trench influence, it influences the orientation of the faulting. Thanks very much.
Geologic research in the Death Valley region of Eastern California has led to numerous and widely applied models for how the continental crust extends. Two prominent models, the rolling hinge model and the discrete normal faulting model, have distinct expectations for the pattern of normal faulting and associated tilting across the region. For my thesis, I worked to constrain the late Miocene tilt history of the resting Spring Range, which is located 50 kilometers east of Death Valley, and to interpret its tilt history in the context of these extensional models. By mapping the Miocene sedimentary and volcanic rocks, dating the volcanic succession using argon-argon geochronology, and conducting paleomagnetic analysis on one volcanic unit, we found that approximately half of the normal faulting that produced tilting in the range occurred between 11.6 and 9.8 million years ago, while half of the tilting in the range occurred after 9.8 million years ago. These results, when considered in the context of other constraints on fault timing in the region, is most consistent with the discrete normal faulting model.
Hi, my name is Samantha Garvin, and my project is an examination of habitat quality in urban parks, specifically looking at cavity nestor abundance and tree regeneration rates in the Bronx Zoo. With point counts and habitat surveys, we found that there are 13 species of cavity nesting birds, and over 77% of all trees found in the park or surveyed in this study were between 0 and 15 centimeters diameter at breast height. A majority of these trees are not utilized by cavity nesting birds, and the park management should adjust their management strategies to compensate for this imbalanced tree age structure. Thank you.
Hi, I'm Leah Pollak. Now, in most songbirds, like you see here, males sing more complex songs than females because they're competing with each other for mates and territory. However, in cooperative breeding birds such as superb starling, which I studied, there are helpers at the nest to help breeding birds raise their offspring. So females often compete with each other for not only mates, but also for helpers to help them raise more offspring. So I wanted to test in this bird whether in a cooperatively breeding bird you would see more complex song in females because they're competing with each other for helpers at the nest. So what I did was I created these visualizations of starling songs that I recorded in Kenya. You can see over there on the left. There's these different kinds of syllables that you can see when you visualize the song. And I measured the number of different syllables each bird could produce and also how diverse their song was. I found that both males and females had equally complex song, and the birds that were breeders, that produced offspring, had more versatile song than the birds who helped at the nest, which suggests that song is a means for both male and female birds in the species to compete for social rank and for the access to helpers at the nest. Thank you.
Hi, my name is Martha Villalba, and my thesis is on assessing populations of painted turtles in ponds of black rock forests. Painted turtles are one of the most studied species of turtles in the world. However, there have been no recent studies done on populations of turtles in ponds of black rock forests. For this reason, I chose to examine the populations in 3 of the forest's ponds. In order to do so, I used a hoop trap, as you can see in the picture, to capture turtles. Turtles were then marked, measured, and recaptured daily during the summers of 2010 and 2011. Using the data I gathered, I used the market capture method to estimate the population size in each of the 3 ponds. I also determined the sex ratio and age structure. My data suggest, when compared to data from the last study, that the populations appear to be growing in all 3 ponds. This finding, as well as my other findings, suggest that the populations are growing and appear to be healthy. Thank you.
study came out a few years ago that showed higher fruit pulp densities higher up in tree crowns, and I expected this to affect the feeding behavior of blue monkeys in Kenya. What I expected is the situation you see in this diagram, where monkeys, when feeding alone, feed towards the top of trees and have high intake rates and low movement rates. And when feeding with others, the dominant individuals stay towards the top, and subordinates are forced to move down in the tree and lower their intake rates and increase their movement rates. What I found was no rank effect, but that intake and movement rates do vary with tree height, which is interesting because it may say something about the costs and benefits of aggressive behavior in this species. Thank you.
Hi, my name is Leah Pollak, and I studied female dispersal in the cooperative breeding bird superb starling. Using microsatellite markers, I found genetic evidence that about 16 to 28% of females immigrate with their sisters, creating what I call a kin substructure, or small pockets of relatives within a larger social group with which they are unrelated, as indicated by the circles in my cartoon. But what's really unique and exciting to my study is that not only were immigrant females immigrating in at the same time within the same dispersal event, but they were also immigrating into a group where a sister had previously established herself. And so this is the first evidence of kin recruitment amongst immigrant females in cooperative breeding birds.
Hi, I'm Kaitlyn Dean, and I also studied superb starlings in Kenya. My study was on the relationship between acacia ant aggression and superb starling nest site choice. And so what that means is I looked at in the area I was studying, there was one dominant species of tree that all the nests were found in, and there were 4 mutualistic ants that lived in this tree. And so I looked at the aggression of those 4 species of ant, ranked the aggression, and then found that the birds were preferentially nesting in the intermediately aggressive species of ant, which suggests that either there's some trade-off going on between acacia ant aggression to predators of birds, or perhaps that there's another variable at play in terms of the way that the ants affect the structure of the tree, pruning behavior or something else that's going on.
Good afternoon. My name is Allie, and I studied carbon accumulation rates in Arctic and subarctic peatlands in Alaska throughout the Holocene period. Peatlands are large and efficient carbon reservoirs and are especially prevalent in the northern hemisphere. Currently, subarctic peatlands experience higher productivity levels than do Arctic peatlands due to higher temperatures and increased moisture levels. In this study, I conducted loss on ignition sampling on sediment from 5 locations in Alaska in order to determine carbon accumulation rates. We found that higher accumulation rates correspond with cooler, wetter climates, indicating that moisture is a key factor in the ability of peatlands to store carbon. We believe that the current functionality of subarctic regions may serve as a proxy for Arctic regions in the future, in that higher temperatures may cause increased moisture via the melting of permafrost. Subarctic regions may experience drying with these increased temperatures, and therefore may become sources of atmospheric carbon and potential sources of concern. Thank you very much.
Good afternoon, I'm Greg Pendergast. This past July, I studied 2 native shrub species in the northern part of Alaska. Warmer winters have led to an expansion in range of both dwarf birch, Betula nana, as well as dwarf willow, Salix arctica. And I wanted to look at how a leaf's position in the canopy will affect its respiratory response across a temperature gradient. So we set up light sensors throughout a canopy, shown by those little hexagons, and then sampled leaves nearby them and subjected them to infrared gas analysis in a lab. And what we found was there was considerable variation in both light environment and respiratory activity in the canopy, but we were surprised to not find a direct correlation. The implications are that big estimates of terrestrial carbon cycling should take a more nuanced approach that include interleaf variation amongst the canopy. Thank you.
Hi, my name is Heather DAngelo, and I'm working with Dr. Krista McGuire, studying the impact of oil palm plantations on soil microbial communities in Malaysia. Malaysia currently experiences some of the highest rates of deforestation in the world, which are driven by the expansion of oil palm plantations. And although we know that deforestation drives biodiversity loss across a wide range of species, this disturbance on soil microbial communities has not yet been fully evaluated. So in order to do so, soil cores were collected from a primary forest or generating forest and an oil palm plantation, and then compared using an assay for microbial biomass and community composition. And our results indicate that microbial biomass is significantly lower in the oil palm plantation, and that microbial community composition is distinct in the oil palm plantation, which suggests that oil palm plantations are experiencing a decrease in ecosystem functioning, and there's lots of future research, and if you're interested, please talk to me. Thank you.
Hi, my name is Nina, and my thesis topic is an analysis of microbial function in soils from rainforest and oil palm plantations in Paso, Malaysia. My research mentor is Krista McGuire. To examine the effects of deforestation and conversion of forest to oil palm plantations, we studied soil microbial communities in and around the Paso Forest Reserve in Malaysia. At Paso, there is a mosaic of primary forest, regenerating forest that was logged in the 1950s, and oil palm plantation that has been in cultivation for the last 25 years. Microbial function in soils and leaf litter were measured using extracellular enzyme assays that determine enzymatic activity in our samples. Oil palm soils had the lowest enzyme activity for enzymes that are relevant for the nitrogen, phosphorus, and carbon cycles. And for results here, you can, which is for acid phosphatase, an enzyme representative of the phosphorus cycle, you can see that oil palm soils have a lower activity. This lower activity indicates that conversion of primary forest to oil palm plantation has negative consequences for nutrient cycling ecosystem function. Thank you.
Hi, my name is Sophie Heissen, and I studied changes in plant and invertebrate communities on green roofs over time and between native vegetation designs. I studied a comparison between the 1-year-old Barnard roof and the 4-year-old roof at Fieldston School. Overall, there is a general decrease in survivorship of plant species over time. However, of the 16 species that were planted, only one was lost, which is pretty great considering that no native plants were thought to be able to survive on the conditions on green roofs. The insect communities followed the vegetation communities pretty closely, decreasing over time in diversity. We found many different types of functional groups of insects, including the decomposers like Columbella, herbivores and pollinators like Diptera and Hymenoptera, and insectivores such as the spiders. The presence of these different functional groups is a positive indication that green roofs could possibly be used for increasing invertebrate and plant diversity over time.
My name is Rebecca Rohr, and I studied plant colonization ecology on New York City green roofs. Most previous green roof work has focused on physical ecosystem services like hydrology and heat flux, rather than on the dynamics of plant community ecology. I set out to better understand how green roof plant communities are influenced by the vegetation in their surroundings. In summer of 2011, I collected biomass and seed data from spontaneous colonizers, weeds, on 10 experimental green roofs throughout the city. I analyzed the patterns of seed inputs and biomass across roofs and conducted an ArcGIS spatial analysis to compare the percent vegetated area in each roof's surroundings to seed inputs. The significant differences of biomass and seed counts across roofs suggest that environment influences green roof plant composition as opposed to on-roof variables. Further, the positive relationship between seed inputs and percent vegetated area shows that surrounding plant communities shape the plant communities of extensive green roofs. These results suggest the potential of colonizers to improve ecosystem services from green roof infrastructure. Thanks.
Hello, my name is Tolly, and my research uses a web of sustainability to assess the trade-offs for human well-being and agrodiversity of smallholder cash crop production. Using data from the Millennium Village of Bansaso, Ghana, I analyzed differences in the levels of health, gender, species density, and species richness as indicated above. Results indicate that diet diversity is higher in cash crop producing households due to significantly increased consumption of alcohol, tea, and coffee. Cash crop farmers also experience increased levels of food insecurity, indicating potential gaps in the savings and investment mechanisms of the village. Gender played no significant role across most of the metrics. However, nutrition was significantly lower for cash crop producing households. Finally, species richness was higher, indicating that cash crop farmers had stable levels of social and ecological resilience. These results are important in guiding the policy for regulation, infrastructure, and education investments in both the Millennium Village as well as areas around the world experiencing the transition from subsistence agrarian lifestyles to cash crop producing market-based economies. Thank you.
Still in Africa today, the caloric and economic needs of many people are not being met through agriculture. As we look to change this, we need to be aware of the sustainability of the methods that we employ. Nitrogen inputs are key to producing more food. However, a byproduct of adding nitrogen fertilizer to the soil is the production of N2O, a greenhouse gas which exacerbates global warming, and NO, a smog precursor which is harmful to both human health and plant growth. I took a field study completed by my advisor which determined this relationship between fertilizer input and emissions output and applied it to survey data of actual fertilizer use by farmers in Kenya, both before and after participation in a Millennium Villages program that encouraged fertilizer use. According to my projections of what emissions might look like, increased fertilizer use resulted in increased emissions, as you can see on the left. However, because fertilizer use also increased food production, emissions per kilogram yield decreased with increased use, as you can see on the right. This suggests that fertilizer use, at the level seen here, is an efficient method of increasing crop yield in terms of its global warming potential. Thank you.
Good afternoon. My name is Jordan Holler-Smith. Vessel groundings are one of the major threats facing coral reefs today. These can lead to a complete destruction of the reef system. One of the proposed solutions, and the subject of my research, is to grow coral fragments in nurseries and plant them out into the affected reefs. Thank you. Hi. My research looked at the species distribution of the phytoplankton genus Sudnicha on George's Bank during the summer of 2010. Sudnicha produces a neurotoxin known as domoic acid, and domoic acid can accumulate in shellfish, which can cause great harm and even death to marine mammals, birds, and humans that ingest them. So for my research, I used molecular approaches to determine the species composition of Sudnicha on George's Bank, and I used a domoic acid ELISA to determine the domoic acid concentrations on George's Bank. What I found was that there were 2 toxic species of Sudnicha on George's Bank, and that each of these occupied its own ecological niche. Also, I found that there were low levels of domoic acid throughout George's Bank during the summer season, but that the highest concentration of domoic acid occurred in June. And finally, what I found was that there was domoic acid present in samples containing neither toxic species, suggesting that one of our non-toxic species may actually be toxic. Thank you.
Hi, I'm Candace Henry, and for my thesis, I assessed the transuranic component in reactor-derived releases from Fukushima Daiichi into the Northern Pacific. As you may know, in March of 2011, the combination of an earthquake and a tsunami caused the shutdown and overheating of Fukushima nuclear reactor in Japan. Fission products such as cesium-134, 137, and iodine-131 were detected in the vicinity. However, little is known about the transuranic component of the releases, such as plutonium and neptunium. Through ICP-MS, gamma spectroscopy, and an N-member mixing model, we are able to determine the amount of Fukushima-derived plutonium in our water samples. From this, we are able to estimate that 8 × 10⁹ becquerels of plutonium was released into our study site. And while this is only 4 × 10⁵% of total plutonium in the North Pacific, the detection of plutonium tells us that unintentional releases of reactor materials did occur. Thank you.
Hi, I'm Susan Fan, and for my senior thesis, I wanted to study the potential impact of natural oil seeps in the Gulf of Mexico on surface phytoplankton communities. Now, previous laboratory studies had differed on whether low concentrations of oil inhibit or stimulate phytoplankton growth. For my research, I chose to use satellite data, which has the advantages of broad spatial and temporal coverage. I constructed a time series of local estimated chlorophyll concentration from around a major seep site in the Gulf and from around 4 control sites that had not seen any slicks between 1998 and 2007. And when compared against a database of satellite-detected oil slicks from the same decade, I found that, on average, after slick events, the seep site experiences a statistically significant increase in chlorophyll compared to the 4 control sites. And this result can be a step towards understanding the impact of man-made oil spills on the planktonic food web. Thank you.
Every major environmental crisis of the 20th and the 21st centuries is linked to the history of industrial pollution. My project is a case study on the industrial pollution and development of the platinum group element industries in New York Harbor over the course of the 20th century. The platinum group elements were used in a variety of industries, ranging from the petroleum industry to the chemicals industry to the automobile industry and the primary metals industry, as you can see in the picture on my right. In the production of a variety of metals, the platinum group elements were utilized and released as effluent into New York Harbor. During the time period from 1910 to 1982, I was able to reconstruct the history of the platinum group elements in New York Harbor. As you can see in the figure on my right, we were able to reconstruct the concentration of the platinum group elements with depth in the sediment. And then we were able to compare these to a variety of dating methods. 2 of the most important included radiometric dating through cesium-137 and also the production history of the platinum group elements themselves. In addition to reconstructing this pollution history, we were also able to provide evidence for the construction of a new geochemical dating tool for industrial sediments across the globe because platinum group elements were used in a variety of industries. This is the first evidence of this dating method that has been produced, and these are the first measurements of the platinum group elements that have been recorded before 1950. Thank you.
Hi, my name is Stephanie Gerberg. The accumulation of discarded plastics represents a global waste management problem. For my thesis, I incubated polyhydroxyalkanoate, a biodegradable plastic, in solutions of common fungal enzymes and monitored the changes in their physical and chemical changes over time, over a period of 5 weeks, using spectroscopy and atomic force microscopy. I found that although the spectroscope that I used did not detect any changes, the atomic force microscope detected changes in surface roughness, thickness, and adhesion, suggesting that these common enzymes do have an effect on the composition of the plastic, and also suggesting that it may be possible to achieve increased degradation using living organisms rather than enzyme solutions. Thank you.
Good afternoon. My name is Thomas Timberlake. The country of Liberia in West Africa suffered from an intense period of conflict throughout the 1990s and early 2000s. I've spent the past year researching connections between this conflict and water access in Liberia. I've used GIS and spatial analysis to look at several indicators of this connection, including distance between water access points and conflict locations, as shown on that graph. The main finding that many of my results pointed to was the heavy influence of population density on the spatial distribution of data in Liberia. Thank you.
Hi, good afternoon. My name is Vasily Hristides, and the main question we sought to unravel is, what is the relative impact of perceived corporate sustainability on stock price performance? And our methods consisted of gathering mean monthly returns over the period 2006 to 2012 for 2 of the leading sustainability rankings, as well as for the S&P 100, which represented conventional firms as a means of comparison. And our findings were that over this entire 6-year period, the sustainable firms significantly underperformed their conventional counterparts. But more interesting is if you look in the 2 years leading up to the 2008 financial crisis, you see that the behavior of these two types of firms was virtually indistinguishable. And only in the years following did the sustainable firms begin to underperform. Now, we believe that this has something to do with increased risk perception, as well as greater idiosyncratic risk for sustainable firms. Thank you very much.
My name is Emily Simone, and I researched whether there is an impact on comparative market valuations for public companies that can be attributed to ecosystem services usage. The benefits derived from ecosystem services are either unpriced or undervalued, so it is unclear what the effect of ecosystem services usage is on stock performance. So results might indicate whether there is an advantage reflected in stock performance to relative efficiency in the usage of ecosystem services or corporate sustainability measures. I used OLS multiple linear regressions to compare a market valuation ratio, price to book, against return on equity, which is a measure of profitability, and ecosystem services intensity measures such as greenhouse gas emissions over dollars of earnings and water usage over earnings. And I found that there is an advantage to efficiency in greenhouse gas emissions that's reflected in a higher price to book value, and there is an advantage to a higher percentage of total assets being represented by land ownership reflected in price to book. Thank you.
Hello, my name is Erin Stommer, and I researched the impact of Hurricane Irene through the creation and testing of flood vulnerability indices in the Northeast United States. The social and capital asset indices I created in ArcGIS were applied to 3 study regions: the Catskills, New York, Northeast New Jersey, and Eastern Vermont. I used Goodman and Kruskal gamma correlation tests to determine patterns of vulnerability and found that across all 3 study regions, socioeconomic status was the primary driver of composite social vulnerability. In the Catskills and Northeast New Jersey, a significant positive correlation was found between capital asset vulnerability and race and ethnicity indicators, highlighting the need to target disaster preparation to minority populations living in crowded areas who may not speak English well. Linear regressions with proxy data of damage caused by the hurricane did not match predicted vulnerability. Thus, more complete proxy data as well as further correlation testing are needed before government officials can use these indices to allocate resources in a cost-efficient manner. Thank you.
Hi, my name is David Solomon. Currently in New York City, 72% of the electricity used is consumed in buildings. In order for New York City to reduce its greenhouse gas emissions, it therefore needs to optimize and reduce energy consumption in existing buildings. For my study, I examined 2 iterations of a model that predicts energy forecasts for commercial buildings. The 2 different iterations of this model are different in their data configurations. The first model, Model A, uses continuous historical energy and weather data. The second model uses non-continuous historical energy and weather data. This means that it's specific to one day of the week. So rather than Monday, Tuesday, Wednesday, Thursday, Friday, it is just Monday after Monday after Monday. As you can see in the figure, Model B, the non-continuous model, which predicts for one day of the week, based on a specific day of the week's historical data, performed better than Model A, which was built using historical continuous data. The high accuracy of this model has implications for operations management in commercial buildings, suggesting that they will be able to predict and prevent excessive energy use, and it also has implications for dominant modeling methodologies such as the DOE 2.2 model, suggesting that it should take a use phase approach in addition to its pre-existing methodology. Thank you.
Hello, my name is Matt Palmer. I'm one of the advisors in the program, and Martin and I are going to finish out the last couple of 1-minute slides for some students who finished their theses in December and aren't able to make it today. So this work was done by Maureen Stamola, a senior in the environmental biology program, studying the evolution of lizards in the Caribbean. She was particularly interested in how a group of related lizards that lived along an altitude transect from hot low elevations to cool high elevations responded to environmental change. And what she found was that, that lizards that live high on the mountaintops are better able to withstand cold temperatures than lizards below, suggesting there was some matching between the physiological capacity of the organisms and their thermal environment. She found this was true both when she compared across groups that were specialists and when looking at a generalist lizard found throughout that range.
So Iceland uses a lot of renewable energies sources, mostly geothermal energy. But while the energy sources are renewable, they still emit CO2 because the groundwater in Iceland has a lot of CO2 in it. So Iceland is very interested in actually storing that CO2 someplace that it doesn't go into the atmosphere. And Bailey worked on a CO2 sequestration pilot study in Iceland where we injected a tracer, SF6, and we will inject other tracers in the future into the subsurface to follow the CO2, how it migrates into the subsurface. And the curve that you see on that graph is a breakthrough curve of SF6 in a monitoring well. That means it tells you how fast the trace and the CO2 arrive at the monitoring wells. And the result of the study showed that the movement of groundwater in that area is very, very slow. It's only about a few centimeters per day or meters per year. Thank you.
Camille Salcedo-Watson traveled to New Zealand last summer to study the effects of an invasive fungus on the native fungal communities living in forest soils, with the expectation as to whether or not that invader reduced the diversity of the native community, to think what might be the consequences for forestry and other sorts of ecosystem services. What she found was that in certain of these transects, we've got a photograph of the forests and the fungi and the fungal root tips to your left. In the data figure, you see that on 2 of the transects, there was a reduced diversity in the cores that contained the invasive fungus, but in 3 other ones it did not. So overall, there did not seem to be an effect of invasion on the native diversity. That could be because the invading fungus was of a functionally different type than the resident fungi. But 2 other interesting findings that popped up were that most of the fungi that she found were undescribed. So there's an enormous amount of undescribed fungal diversity. You know what? I forget the second interesting thing, but it's in her poster, and I'll be happy to talk to you about it when we're over there. Okay, and the last slide will be Miguel Macias, who is studying the physiology of respiration in oak leaves along an urban-to-rural gradient. The idea here is that trees may become adapted to the local environment, and urbanization is one of the major environmental changes. To what extent is tree physiology adapting to that? So what he did is he collected tree leaves and he exposed them to increasing temperatures in the laboratory and looked at their physiological capacity. And he found fairly interesting but hard to interpret results, which were that the thermal capacity, the efficiency of the photosystems varied such that the efficiency was actually highest in the urban environment, but there wasn't a consistent trend across, and that the critical temperature, which was the temperature at which photosynthesis basically begins to fail, was highest in the suburban environment rather than the urban. So there were not clear linear trends with either of those, but it did suggest that there was some site-specific adaptations to physiology in these trees that probably will play into the long-term effects of carbon sequestration.
So it's time for a big round of applause, I think. So I think these presentations hopefully convinced you what a great job undergraduate students can do in research projects. If you want to have one of those students, next generations of students to work with you in the future, come see me. I can put you in touch with people that can make that happen. I also forgot a couple of people in my acknowledgement, so I'd like to thank Catherine Cook and her many student helpers in the environmental science department, for guiding us along during the semester, Barnard Facilities for putting this all up, and the Barnard Media Department to take care of the video and everything else technologically today.